Today, I'm going to talk about some of the work that we've been doing to establish animal models that will hopefully support the development of psychedelic-based therapeutics. So psychedelic drugs that are often described as powerful psychoactive substances that can alter perception, mood, and cognition have been used by humans for many thousands of years. Our research relationship with this class of drugs likely began in the 1940s with the accidental discovery of the psychoactive effects of LSD by Albert Hofmann. So one day after working in the lab, Hofmann describes feeling like he was in a dream-like state and seeing intensive colors surging towards him. Some hours later, when the effects dissipated, Hofmann concluded it was likely an accidental exposure to LSD that produced these effects. So he did as any good scientist would do and exposed himself to some more some days later. And here it appears on his second exposure, he had taken a higher dose where he describes losing all control of space and time and experiencing quite intense outer body experiences. So for the title of my slide here, I've paraphrased what's often said about psychedelic drugs, that they're the oldest and least understood psychopharmacological agent. And I think Hofmann's description of the effects of LSD goes some way to show how little we understand about this class of drugs. I think most neuroscientists will agree that Hofmann's experiences are not well understood on a biological or pharmacological basis. And this is likely due to a lack of research into this class of drugs. So after the change in the legal status of these drugs in the 1970s, we have quite a barren period for research publications on psychedelic drugs. Thankfully, in recent years, we've seen a resurgence in interest in psychedelics. And this is likely due to the discovery of the therapeutic potential of these drugs in psychiatric diseases. And we have seen many clinical trials registered focused on psychedelic drugs, in particular psilocybin, and different psychiatric conditions in recent years. I think arguably the most exciting discovery so far to come from this recent resurgence in psychedelic research was published earlier this year. And this shows that psilocybin performs at least as well as a standard antidepressant drug, escitalopram, at reversing depressive symptoms in patients over a 6-week period. Now, this is a fascinating discovery on many levels. One being that antidepressant drugs with a novel mechanism of action aren't discovered very often. And secondly, psilocybin required only 2 days of dosing within this trial, whereas escitalopram required daily dosing for the 6-week period. So I think there is quite some promise in the therapeutic potential of psilocybin and other psychedelic drugs for depression, but also potentially for other psychiatric disorders. So I've mentioned a couple of different psychedelic drugs so far. Before I go any further, just a quick note on their pharmacology. Now, this is by no means a comprehensive overview of all psychedelic drugs, nor is it a comprehensive overview of the pharmacology of these drugs. I think we still have a lot to learn from this going forward. But here I've listed some commonly known psychedelic drugs. So LSD and DOI, which are synthetics, DMT, which is initially discovered endogenous in plants, but also is endogenous in mammals, and psilocybin, which I've already mentioned. But here I've shown the pharmacology for its active metabolite psilocin, which is likely responsible for most of the psychedelic effects or most of the psychoactive effects of psilocybin. So as you can see, the pharmacology of these drugs can be quite diverse. What they mostly have in common is activation of 5-HT2A and 5-HT2C receptors. And in fact, it's thought that activation of 5-HT2A is likely responsible for the psychoactive effect of these drugs. Just a quick note on other drugs that are often discussed under the heading of psychedelics, and this is dissociative anesthetics. These differ quite considerably in their pharmacology from the classic serotonergic psychedelic drugs, in that they act as NMDA receptor antagonists rather than on serotonin receptors. But they do produce similar effects in that they have perception altering abilities. So today I'm going to focus primarily on psilocybin and DOI, But I will also mention ketamine because I think it shows similarities to psilocybin, at least with respect to the treatment of depression. So as I mentioned, today I'm going to cover the work we've been doing to assess the suitability of different animal models to support psychedelic-based drug discovery. There are quite a number of ways in which animal models are essential for the drug discovery process. I've listed 4 here. What I'm going to focus on today is target engagement and preclinical efficacy testing in relation to depression models. So when I say target engagement, what I mean is in vivo target engagement. So can a drug, in the case of psychedelics, penetrate the brain, activate the receptor of interest, and produce the desired effect? And I think as a field, we're well placed to assess target engagement of psychedelic drugs, particularly in relation to activation of the 5-HT2A receptor. And this is thanks to this publication in Science from 1956 that reports a rapid and violent head shaking in mice following administration of LSD. And the authors note that this is something that's not seen in animals that haven't been administered a psychedelic drug. So it's not a normal behavior. And this publication paved the way for the use of the head twitch response over the years and up until present day to identify psychedelic drugs or drugs with psychedelic-like properties. So here I show some data that we have generated using the psychedelic DOI, where we see a dose-dependent increase in head twitch counts in mice. Now, as our understanding of pharmacology progressed, it was found that the head twitch response was actually a 5-HT2A dependent response. And this paved the way for the use of this response beyond discovering psychedelic drugs. And that was in the identification of 5-HT2A receptor antagonists, of which many atypical antipsychotics are. And here I show some data from the work we've done around a couple of atypical antipsychotics, where we can assess the potency of these drugs as 5-HT2A antagonists in their ability to, by measuring their ability to block DOI-induced head twitches. And this is an assay that we do routinely use to identify 2A receptor antagonists. But we have seen a resurgence in the use of the head twitch response in recent years with regards to identifying and studying psychedelic drugs. So here I show some data from a small study that we've carried out in mice using psilocybin and the head twitch response. I think a key question for the field of psychopharmacology and behavioral pharmacology currently is what's the optimal dose of psychedelic drugs in rodents, and what is the duration of action? And as you can see here, the head twitch response can be quite useful here for dose ranging studies and for studying the duration of the effects of different psychedelic drugs. Now, although we are very well placed here to study target engagement using the head twitch response, it arguably tells us very little about the efficacy of these drugs in disease models. When we think about efficacy testing in depression, what is probably the most widely used method of assessing antidepressant efficacy in rodents is the forced swim test. Just a quick description for those of you that aren't familiar with this. Essentially, a rat or a mouse is placed in an inescapable cylinder of water, and their behavior is studied over a short period of time. The main behavior that is measured, or perhaps the most significant behavior, is immobility behavior, which is the time in which the animal is not actively trying to escape from the cylinder. And the significance of this immobility behavior is that it's interpreted as a depressive-like behavior, either in the form of behavioral despair or learned helplessness. And what's important about this is that antidepressant drugs reduce this immobility behavior in rats and in mice. And I show an example of 2 antidepressant drugs here that are on the market, fluoxetine and reboxetine, and both significantly reduce immobility behavior. Now, while talking about the forced swim test, I think it's worth addressing some of the criticism of the test, of which there is quite a lot, some of it well-founded. I think we often question what is this immobility behavior? Is it behavioral despair? Is it learned helplessness? Is it measuring anxiety? Is it measuring cognition? Or is it measuring anything useful at all? What I will say is that immobility in the forced swim test has proven to be a useful pharmacodynamic readout of antidepressant drugs, particularly monoamine reuptake inhibitors. So we have thousands of publications in the literature over the past few decades, consistently showing, for the most part, that antidepressant drugs reduce immobility in the forced swim test. So I think it's only natural that when researchers are developing a new antidepressant drug, it's only natural that we want to compare these new drugs to what's previously been shown with antidepressant drugs that are currently on the market. And immobility in the forced swim test is very useful for this. But I think one important question about the forced swim test is, and this immobility behavior, is it useful for screening antidepressant drugs beyond monoamine reuptake inhibitors? And this is a question that we as a field have asked before. And this is in relation to antidepressant drugs with a novel mechanism of action, particularly around glutamatergic pharmacology. I think many of you will know that ketamine has recently, or specifically S-ketamine, has recently been approved for the treatment of depression. And as I said earlier, I think ketamine and S-ketamine in the clinic show similarities to psilocybin in that a single dose is sufficient to induce reduction in depressive symptoms in patients. But before the approval of ketamine and the clinical trials, a lot of preclinical work was done focused on studying ketamine in rodents, and particularly in the forced swim test. And one key question that did come up some years ago was the forced swim test useful for studying ketamine and other glutamatergic or ketamine-like antidepressant drugs? And the answer to this question was yes. So here I show some data that we generated some years ago, but published earlier this year, focused on investigating ketamine in a rat model of depression. And what we see is that ketamine does significantly reduce immobility behavior with a rapid onset of action and a longer duration of action, which is in some way similar to what we see in the clinic. But I think one key question for psychedelic research is the forced swim test useful for studying psychedelic drugs that may show potential for the treatment of depression? And I think the jury is very much out on this at the moment. So here I show some data that we have generated using psilocybin in the mouse forced swim test. And so far, using these doses, at least at these time points, we have seen no effect of psilocybin in the mouse forced swim test. There's also been some publications on ketamine in the mouse forced swim test, or sorry, on psilocybin in the mouse forced swim test, which has investigated slightly different doses and slightly different time points to what we have done, but it does go some way to back up what we've shown in that psilocybin is not efficacious in the mouse forced swim test. In contrast to this, psilocybin and LSD, in fact, have been shown to induce persistent antidepressant-like effects in the rat forced swim test. And these effects do last for several weeks after administration, which may show some similarities to what we see with psilocybin in depressed patients in the clinic. Now, I think we're at a very early stage on answering the question as to whether the forced swim test is going to be useful for psychedelic research. But I think that there is certainly some questions to be asked around this, and it's something that we are actively researching at the moment. So hopefully we will come close to an answer on this quite soon, because I think the forced swim test still can show some utility for screening novel psychedelic drugs. But in the meantime, we are focused on developing some assays, some behavioral assays that are more similar to the symptoms of depression that are reported in the clinic. And one thing that we have focused on is motivation. And this is to tie in with the symptom of depression that's quite well known as anhedonia, which is the inability to feel pleasure in normally pleasurable activities. Now, anhedonia itself can be subdivided into motivational and consummatory anhedonia. What we've focused on is motivational anhedonia. So it's at least in the clinic described as a loss of interest or an absence of anticipatory pleasure. And the reason we focus on this is that motivation can be quite easily measured in rodents using the progressive ratio task. This is an operant-based test of motivation. So as a quick description of the test, the rat is placed in this operant conditioning chamber and trained to press a lever to receive a reward in the form of a sucrose pellet. Now, how this task works is that for every subsequent reward the animal receives, it must press the lever more times. And from this, we can get a measure of total lever presses over a session, or break point, which is essentially the time in which the animal gives up. This task is known to be dopamine dependent. So dopamine antagonists or dopamine depletion reduces responses and break point in the task. We have also investigated inflammatory inducing stimuli in the task. So when we administer LPS, animals reduce their break points and lever presses acutely, which is during the phase in which the animals will be experiencing sickness behavior induced by LPS. But what's perhaps more significant is that at the 24-hour time point post LPS administration, which is well established to be beyond sickness behavior, but the time in which rodents show depressive-like behavior, we see a reduction in breakpoint and lever presses, which suggests that this task is useful for studying depression-inducing stimuli. So we wanted to use this test to initially study the effects of psilocybin and other psychedelic drugs on motivation in rodents where we haven't induced anhedonia. And psychedelics have been investigated in this task previously. So I show some data here from a good friend and collaborator of ours, Guy Higgins, based at Intervivo Solutions, who has earlier this year published the effects of microdoses of psilocybin on the performance in the progressive ratio task. And what they've shown is a dose-dependent increase in motivation with microdoses of psilocybin. And specifically, this is seen only in animals that show a low level of motivation at baseline. So this may well have implications for anhedonia. So we've taken a slightly different approach in that we have examined macro doses or at least doses that induce these 5-HT2A dependent behaviors that I spoke about earlier in rodents. So here I show on the top panel breakpoint, on the bottom panel lever presses. Vehicle is here in black, and I have 2 doses of DOI in blue and in green. And if we look at the high dose of DOI here, we see acutely there's a significant reduction, which is quite robust in breakpoint and lever presses. And this is 30 minutes post-administration. Now, I think you can probably question the utility of measuring these outcomes, these motivation outcomes, when the animal is displaying these 5-HT2A dependent behaviors, which I think is certainly a well-founded question. But what we actually wanted to focus on were the longer term effects of psychedelic drugs. And here we see actually a significant enhancement of motivation in the days following DOI administration. And these effects become apparent around day 4 post-administration and persist to day 6. Now, DOI is as far as I'm aware, not currently under clinical development for the treatment of psychiatric diseases, and is probably unlikely to be so. So what we wanted to investigate here was a drug that is well established to be efficacious clinically in the treatment of depression. So I think at this point, the gold standard in that regard is psilocybin. And here we investigated, again, 2 doses of psilocybin that would be sufficient to induce these 5-HT2A dependent behaviors. Again, I show vehicle and a medium and higher dose of psilocybin. And interestingly, what we see here is almost a complete replication of what we see with DOI, in that we see a significant and robust decrease in motivation acutely, followed by a prolonged enhancement in motivation, particularly at the higher dose of psilocybin. And here, in fact, we actually likely are seeing a more potent effect than DOI, in that what we see is apparent, again, 4 days following psilocybin administration, but this does persist to at least day 7. And in fact, we have followed this up with subsequent experiments, and we are seeing effects until at least day 14, which is interesting because this, again, probably in some way replicates the time course of effects that we see with psilocybin clinically with respect to depression, but also the time course of effects that are now being published in the literature, showing the molecular and synaptic changes following administration of a single dose of psilocybin. Now, finally, as I've said, we can actually stratify animals based on low and high motivation at baseline. And this is what we've done here using DOI, using the highest dose of DOI, the 1 mg per kg. We have investigated animals that show a low response at baseline and a high response. So animals with a low level of endogenous motivation or an endogenously high level of motivation. And what we see is quite a difference in the response to DOI. So the animals that start with this low level of motivation are not susceptible to the acute reduction in motivation induced by DOI, but do somewhat show the long-term enhancement in motivation. With the high responders, what we see is that animals that these animals show the opposite to the low motivated animals, in that they are susceptible to this robust decrease in motivation acutely, but do not show a prolonged enhancement in motivation in the days post DOI administration. Now, this suggests a potential complex relationship between psychedelic drugs and motivation. It does beg the question as to whether psychedelic drugs can act to correct changes in motivation or reward-seeking behavior, whether that be a low level of motivation in the form of anhedonia that we see in depression, or perhaps a high level of reward-seeking behavior, a high level of motivation that may be seen in other psychiatric disorders, such as addiction. Now, I think we are quite far away from drawing any conclusions on that, but I think it is an interesting question to ask, and it is something that is actually being explored in the clinic currently, as to whether psychedelic drugs can reverse anhedonia or can correct pathological reward-seeking behavior seen in diseases such as addiction. And I think over the next months and years, I think we'll see more and more research being focused on this particular question. Now, I've quoted Albert Hofmann already today. He describes his experiences with LSD as almost a life-changing experience. I think there is quite some research to be done in the form of basic research and drug development, but hopefully as things progress, we will start to see patients with psychiatric diseases describing psychedelic drugs as life-changing experiences in the coming years. Now, finally, I just want to thank all of the Transpharmation team that were involved in this research, in particular, Fionn, who has generated the progressive ratio data, Irvina, Luke, and Sylvia, who have worked on the forced swim and head twitch. I want to thank Kerry for her input into the talk, and Col and the rest of her team who had also generated some of the head twitch data. And of course, thanks to you all for listening.